I'm sure many of y'all have already heard of the molecule DNA, and it stands for deoxyribonucleic acid. I wrote it out ahead of time to spare you the pain of watching me spell this in real time. But it is, and I think you already have an idea of this, is the basic unit of heredity, or it's what codes all of our genetic information. And what I want to do in this video, because I think that's kind of common knowledge, that's popular knowledge, that everything that, what makes my hair black or my eyes blue or whatever, that's all somehow encoded in our DNA. But what I want to do in this video is give you an idea of how something like DNA, a molecule, can actually code for what we are. How does the information, one, get stored in this type of a molecule, and then how does that actually turn into the proteins that make up our enzymes and our organs and our brain cells and everything else that really make us us. So this is a computer graphics representation of DNA, and I'm sure many of y'all have heard of the double helix. Double helix. And that's a reference to the structure that DNA takes. And you can see here, it's a double helix. You can see here, you have two of these lines, and they're intertwined each other. You see there, that's one of them. And then you see another one intertwined like that. And then they're connected by, you can almost view it's like these bridges between the two helixes, and they twist around each other, and I think you get the idea. And so the double helix just describes the structure, the shape that DNA takes, and it leads to all sorts of interesting repercussions in terms of how heredity takes place and how natural selection and variation might take place as well. And actually, in the future, I do want to actually read with you the Watson and Crick's paper on the double helix, where they essentially, talk about their discovery. And the best thing about that paper, besides the fact that it was probably one of the biggest discoveries in the history of mankind, is that the paper is only a page and a half long. And it goes to my general view that if you have something good to say, it shouldn't take you that long to say it. But with that said, let's think a little bit about how this can actually generate the proteins and whatever else that make up all of us. So right here, this is a zoomed up version of that graphic that I just showed you a little bit earlier. And this is each of the helixes. So if, this is the magenta side, if you unwound this helix, right now it shows it in kind of its wound state. But if I unwind this helix, I'd get one side would maybe be this magenta side of our helix. And then one side is this green side, right? And if you twist it up, you get back to this drawing up here. And then these bridges that you see in this drawing, in the double helix, those are these connections right here. These are the bridges. These are the bridges. Now, what allows us to code information is that the blocks that make up the bridges are made of different molecules. And the four different molecules that are made up in DNA are adenine. And it's written here on this little chart. I got all of this from Wikipedia, so if you want more information, I encourage you to go there. Adenine, they do that. That's up here. This is the molecular structure of adenine. It's connected to a sugar right here, ribose. I won't go into deoxyribose. That's where it comes. And then you have your phosphate group. But these kind of form the backbone of the DNA, the sugar and the phosphate groups. And I'm not going to go into the microbiology of it, because that's not important right now for understanding just how does this intuitively code for what we are. So you have, but along the backbone, which is identical, and we'll talk about, they run in different directions. It's called anti-parallel. So if they label the ends, and I'm not going to go into detail there. But the important thing are these bases here. So you have adenine, and adenine pairs with thymine. Adenine pairs with thymine. And you see that up here. If you have an adenine molecule here, on an adenine base here, it'll pair with thymine. This is called the base pair. Adenine and thymine pair with each other. If you have thymine, it's going to pair with adenine. And then you have guanine, and it pairs with cytosine. So guanine, and it pairs with cytosine. And the names of these, you should know these names, just because they are almost well, if you ever enter any discussion about DNA and base pairs, this is, expected knowledge. But the names of the molecules and how they're structured, not important just yet. But what's important is the fact that there are 4 of them, and that they essentially code information. So you can view one of these strands in kind of a simplified way. You could just view it as a strand of, so this one, it has an adenine, and then it has a cytosine, then it has a guanine. It's a guanine, they did it in purple. And then it has it has a thymine, not a guanine. So it has a thymine in purple, and then in blue it has a guanine. So this strand right here codes ACTG. And if you were to code the opposite side of the strand, you could immediately, I don't even have to look here. I can look at this side and say, okay, adenine will pair with thymine, cytosine pairs with guanine, thymine pairs with adenine, and guanine pairs with cytosine. So they're complementary strands. So if you think about it, they're really coding the same thing. If you have one of them, you have all of the information for the other. Now, in our DNA, in a human's DNA, so you might say, hey, Sal, how do I go from these little chains of these molecules? And yeah, I mean, it's like, how does that turn into me? How does that turn into this complex organism? And the simple answer is well, the human genome has 3 billion of these base pairs. 3 billion. 3 billion base pairs. And that's actually just in half of your chromosomes. And I'll tell you maybe in this video or a future video why we only consider half of your chromosomes. And that's because essentially you have a pair of every chromosome. I'll talk more in more detail about that. And this number, to some people, they might say, it only takes 3 billion pieces of, 3 billion base pairs to describe who I am. And to some people would say, wow, it takes 3 billion base pairs to describe who I never thought I was that complex. So depending on your point of view, this is either a large or a small number. But when you take these 3 billion base pairs, you're actually encoding all of the information that it takes to make, in this case, a human being. And actually, it turns out a lot of primates don't have that many different base pairs than human beings. And it's actually what the amazing thing is, even things like roundworms and fruit flies also number in the a surprisingly large fraction of the base pairs of a human being. Maybe I'll do another video where I go into comparative biology. But how does these base pairs actually lead to proteins? I mean, it's fair enough. That's information. It's like you can view these as ones and zeros in some type of computer language. But really, they're not just ones and zeros, because they can take on 4 different values. They can take on, a T, a C, or a G. So you could think of them as 0, 1s, 2s, and 3s. But I won't go into that whole aspect of it just now. So how does that actually code information? So DNA, when it actually transcribes something, the process is called transcription. Let me do it. Transcription. And I'm going to do a pretty gross simplification of it, but I think it'll give you the gist of how it codes for proteins. So what happens when transcription happens is that these 2 strands split up. And one of the strands, let me just take one of them. Let's say it looks like this. I'll do it all in one color. Let's say it's just A, T, G, G, A, C, G. I'm just making up stuff. T, A. Let's say that that's the strand that got split up. And what happens is it transcribes it, and I won't say itself. There's a whole bunch of enzymes and proteins, and it's a whole bunch of chemical reactions that have to happen. But this DNA essentially transcribes a complementary mRNA. An mRNA, and I'll introduce RNA, that RNA, it's essentially the exact same thing as, well, the word is ribonucleic acid. So it's literally, you get rid of the deoxy, so you can kind of say it's got its oxy, and it's ribonucleic acid. But it's very similar to DNA. It codes in the exact same way. And the only difference between RNA, instead of a thymine, it has something called a uracil. A uracil. So every place where you would have expected a thymine, you would have expected a T, you'll now see a U. So for example, if this is the DNA strand, then a RNA, an mRNA, a messenger RNA strand will be built complementary to this. So it'll be built, let's see, with A, you'd normally have thymine when you're talking DNA, but now we're talking RNA. So it'll be a uracil, then an adenine, cytosine. Let's see, uracil. Then we got a guanine, a cytosine, an adenine, and then we'll have a uracil. And so this is the mRNA strand here. And all of this is occurring inside the nucleus of your cells. And we'll do a whole series of videos in the future about the structure of our cells. But I think most of us know that our cells, and I'll talk more about eukaryotic and prokaryotic organisms in the future, but most complex organisms, they have a cell nucleus where we have all of our chromosomes that contain all of our DNA. And so this mRNA then detaches itself from the DNA that it was transcribed from, and then it leaves the nucleus. And it goes to these structures called ribosomes. And I'm oversimplifying a little bit, but at the ribosomes, this mRNA is translated into proteins. So let me do that. So let's say this is the mRNA. It was transcribed from that DNA. So let me get rid of that DNA now. I got rid of the DNA. This is the mRNA that we were able to transcribe from that DNA. And they have these other things called tRNA or transfer RNA. And what these are, and this is the really interesting part. So you may or may not know that, pretty much everything we are is made up of proteins. And these proteins, the building blocks of proteins are amino acids. And for those of you who like to lift weights, I'm sure you've seen ads for amino acid supplements and things of the like. And the reason why they talk about amino acids is because those are the building blocks of proteins. Amino acids. My son actually has an allergy to milk protein, so we had to get him a formula that was just pure amino acids, just all of the milk proteins broken down. So if you look at a protein, it's actually a chain of these amino acids, and usually a fairly long chain. We'll look at some protein structures in the very near future, just to give you an idea of things. It's a very long chain of these amino acids, and there are actually 20 different amino acids. 20 different amino acids are pretty much the structure of all of our proteins. Let me write that. 20 amino acids. Amino acids. So a very obvious question is, how can these things code for 20 different amino acids? I can only have 4 different things in this little bucket right here. And then you just have to go back to your combinatorics, or if you can't go back to it, watch the playlist on probability and combinatorics. You say, okay, there's only 4 ways that each of these that I can have for each of these bases. There's only 4 different bases that I can have here, either an adenine, guanine, a cytosine, or depending on whether we're talking about DNA or RNA, a uracil or a thymine. But how can we increase the combinations? Well, if we include 2 of them, if we include 2 bases, then how many combinations can we have? Well, we have 4 possibilities here, and then we'd have 4 possibilities here. So we'd have 16 possibilities. But that's still not enough. That's still not enough to code for one of 20 amino acids to say, hey, this is going to code for amino acid number 5. And we'll talk more about their actual names. So what do we have to do? Well, we have to use 3 of them. So 3 of them, there's actually 4 times 4 times 4 possibilities here. So they could code for 64 different things. They could take on 64 different combinations or permutations, this UAC right here. So if we have 3 of these bases, we can actually code for an amino acid. Or actually, it's overkill, because we can actually have 64 combinations here, and there are only 20 amino acids. So we can even have redundant combinations code for different amino acids. For example, we might say that, let's say, and this isn't the actual code, but maybe UAC, and I should look these up. This codes for amino acid number 1. And if it was or let's say AAU, this codes for amino acid number 2. And if we have, let me see, if I have, I think you get the idea. If I have GGG, this codes for amino acid number 10. And what happens is, when this messenger RNA leaves the nucleus, it goes to the ribosomes, and at the ribosomes, we're going to look at that diagram in a few seconds, but at the ribosomes, let me take my same mRNA molecule. And they're much longer than what I'm showing here. This is just a fraction of an mRNA molecule. So I'll take my mRNA molecule. And what they do is they essentially act as a template for tRNA molecules. And tRNA molecules are these molecules that are attached to they're almost like the trucks for the amino acids. So let's say I have some amino acid right here, and then I have another amino acid that's right here, like that, and then I have another amino acid that's like that. They'll be attached to tRNA molecules. So let's say that this tRNA molecule has on it, it's attached, so this amino acid is attached to a tRNA molecule that has the code on it A. Let me do it in a darker color. It has the code A U G. And this one right here has it on. It has the code. It has on. Let's see the code. Let me pick another one. Let's say it has G A C. So what's going to happen when you're in the ribosome, and it's a complex situation, but actually what's happening isn't too fancy. This tRNA, it wants to bond to this part of the mRNA. Why? Because adenine bonds with uracil, uracil bonds with adenine, and guanine bonds with cytosine. So it'll pull up right here. It'll pull up right next to this thing. And actually, I should probably, let me see if I can, well, I don't know if I can rotate it. But it'll just pull up right here on attached to this mRNA molecule. And this right here is tRNA. tRNA. This is mRNA. And the names don't matter. I really just want to give you the big picture idea of how the proteins are actually formed. And this is an amino acid. I don't know, let's call it amino acid 1, amino acid 5, amino acid 20. This guy, he's going to pull up right here. The guanine's attracted to cytosine. And if you watch the chemistry videos, these are actually hydrogen bonds that form the base pairs. Adenine wants to pull up to uracil, cytosine to guanine, and so on and so forth. And so once all of these guys have pulled up, let me do that. So once you've pulled up, let's say that this is, I could do it up here. This is my mRNA molecule. I'm not going to draw the specifics right there. My little tRNAs pull up next to it. And they each hold a payload, right? So this first one holds this payload right here of this amino acid. The second one holds this payload of this amino acid, and so forth and so on. And so, it might keep going, and there's another green amino acid here. They really don't have those colors, but I'm just for the sake of simplicity, like that. And then the amino acids bond to each other when they're held like that, close to each other. And this doesn't happen all by itself. The ribosome serves a purpose, and there are enzymes that facilitate this process. But once these guys bond together, the tRNA detaches, and you have this chain, this chain of amino acids. And then the chain of amino acids starts to bend around. So they have all of these, and it's actually fascinating. I mean, people have done many, people spend their life studying how proteins fold. And that's actually where they get most of their structural properties. It's not just the chain of the amino acids, but what's more important is how these amino acids actually fold. So once you fold them, they form these really ultra complex patterns based on what amino acid is attracted to what other amino acids, and these very intricate three-dimensional shapes. And what I took here from Wikipedia is these are some amino acids. And just to be able to relate this to the DNA, this right here is insulin. It's key in our ability to process glucose in our body. So this right here is insulin. It's a hormone. So, sometimes you hear people talking about your immune system. Sometimes you hear people talking about your endocrine system and hormones. Sometimes your digestive system. And this is hemoglobin. Essentially what makes what essentially transports our oxygen in our blood. But all of these things are proteins, and all of these little folds you see, these are all little amino. I mean, there's just little dots of amino acids, and this is just one. Well, some of these are multiple chains of amino acids, kind of fitting together like a big puzzle, but some of them are just single chains of amino acids. For like insulin right here, this is 50 amino acids. And then once the chain forms, it all bundles together and forms this little blob like you see. But the shape of that blob is super important for insulin being able to perform the function that it needs to perform in our systems. But this right here is approximately 50, I forgot the exact number, amino acids amino acids. This right here, this immunoglobulin G, which is part of our immune system, this is roughly 1,500 amino acids. Amino acids. So how much DNA or how many base pairs had to code for this? Well, 3 times as much, right? Because you have to have 3 base pairs that code for one amino acid. And actually 3 base pairs, this is called a codon. Codon. Because it codes for amino acids. So 3 base pairs make a codon. So if you have 50 amino acids that make up insulin, that means you're going to have 50 codons, which means you have to have 150 bases, or 150 of these A's and G's and T's. If you have 1,500 amino acids, that means you're going to have 1,500 codons, which means you're going to have roughly 4,500 of these base pairs that code for it. Now, you might, there's some notions that get confused a lot. So I went to kind of the smallest level of our DNA right here. And this is the level at which, well, this is RNA that I'm pointing to right there. But this is the smallest level of DNA, and that's the level at which the information is actually coded. But how does that relate to things like genes and chromosomes and things that you might talk about in other contexts? And so these base pairs, so let's say the 150 base pairs that coded for insulin, these make up a gene. A gene. So, and these 4,500 base pairs make up another gene. Now, all of the genes don't make proteins, but all of the proteins are made by genes. So let's say I have just a bunch of, let me just take a bunch of, I'm just making another, A, G, and it goes down, 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 and you have, a T and then a C and a C, and let's say I have, 4,500 of these. These could code for a protein. These could code for a protein, or they could have all of these other kind of regulatory functions telling what other parts of the DNA should and should not be coded and how the DNA behaves. So it becomes super, super complex. But this kind of section of our DNA, this is what we refer to as a gene. This is what we refer to as a gene. And a gene can have anywhere, from a couple of hundreds of these base pairs or these bases to several thousand of these base pairs. Now, a gene is that part of our chromosome that codes for a particular protein or serves a certain function. Now, there are different versions of genes. Right? So a gene, so this could, it's a gross oversimplification, but maybe to say this is the gene for insulin. Insulin. Now, there might be, slight variations in how insulin can be coded for, and I'm kind of going out of my domain right here because I don't know if that's true. And maybe I shouldn't just speak specifically about insulin, but it's coding for some protein, but there's maybe multiple different ways that protein can be coded. Maybe instead of a T here, sometimes there's a C there. It still codes for the same protein. It doesn't change it quite enough, but that protein acts just a little bit different. It has a little bit of, it's a slight variant. I'll use that word. Now, each variant of this gene is called an allele. Allele is a specific variant of your gene. Variant of your gene. Now, if you take this DNA chain, and this chain over here, let's see, this is one base pair, and just so kind of, this might be like one base, this is another base, maybe this is an adenine, and then this would be a thymine over here in green. This is an adenine, this would be a thymine. If this is a, if right here this is a guanine, then right here would be a cytosine. This would be just a very small section. If I were to zoom out, right, and let's say we have a big chain of DNA where each of these little dots are a base pair that I'm drawing here. Maybe this section codes for gene 1. Gene 1, maybe. And then there's some noise or things that we haven't fully understood yet. And I want to be clear, just with this simple discussion of DNA, we're already kind of approaching the frontiers of what we know and what we don't know, because DNA is hugely complex, and there's all of these feedback structures, and certain genes tell you to code for other genes and not to code for other genes, and to code under certain circumstances. So it's hugely complex. So there's huge sections of DNA that we still don't understand what exactly they do. But then maybe they'll have another section here that codes for gene 2. Maybe gene 2 is a little bit longer. Maybe it's 1,000 base pairs. But then when you take all of these and you turn it into and then it kind of winds in on itself like this. Let me do it. So it'll wind up winding in on itself like this and do all sorts of crazy things. Remember, this is the whole, it completely bundles itself up, and then it looks something like that. Then you get a chromosome. A chromosome. And just to get an idea of how large a chromosome is compared to the actual base pairs, chromosome number 1 in the human genome. So we have 23 pairs. If you look at it inside of a nucleus, so let's say that's the nucleus. Let's say this is the cell. The cell is much bigger than what I'm showing. But we have 23 pairs of chromosomes. We have 23 pairs of chromosomes. 1, 2, I won't do all of them. And you can actually see chromosomes in a not too expensive microscope. So we're already getting to a scale that we can start to look at. But the largest chromosome, which is chromosome number 1 in the human genome, just to give an idea of how much information it's packing, that thing right there has 220 million base pairs. So when we're talking about, sometimes people talk about chromosomes and genetics and genes and base pairs interchangeably, but it's very important to kind of get an idea of scale. These chromosomes are a super long strand of DNA that's all configured and bundled up, and it contains 220 million base pairs. So the actual elements that are coding for the information are unbelievably small relative to the chromosome itself. But now that we understand a little bit, and actually I want to take a look back at this, because this is what, this kind of blows my mind, that if you just take those little combinations of those amino acids, you can form these very intricate, very advanced structures that we're still fully understanding how they actually interact with each other and regulate how all of our biological processes work. And what's even more amazing is that this scheme that I've talked about in this video, about DNA to mRNA to tRNA to these molecules, this is true for all of life on our planet. So we all share the same mechanism. Me and this plant, we're distant. We share that common root of that we all have DNA. As different as me and that roach that I might not like to be in the same room. We all share that same common root of DNA, and that all of it codes to proteins in this exact same way. That there's this commonality amongst all life. That to me is mind-blowing. Then even more mind-blowing is how these very complex shapes are formed by this, by the DNA. And this isn't speculation. This is, this is observed behavior. That this is a fascinating structure right here. But it's just based on 20 amino acid. You can almost view the amino acid as the Legos, and you put the Legos together, and just the chemical interactions form these fairly impressive structures right here. So now that we know a little bit about DNA and how it codes into protein, we can take a little jump back and talk a little bit more about how variation is actually introduced into a population.